Okay, hi. This is a recording for chapter 16, How Populations Evolve. All right, so just a brief outline of the chapter. We'll talk about genes, populations, evolution. We've already started talking about evolution in our last chapter, more of kind of pretty much mostly Darwin's theory of evolution and natural selection. So now we'll kind of talk a little bit more about natural selection specifically and the different types of natural selection. And also just talk about maintenance of diversity within the population. So again, this is all focused on population. And I actually mentioned this in class as well the other day when we were talking about, and when we covered chapter 15. So an example of, antibiotic resistance. Okay, so MRSA, right? We've heard of that probably before. It stands for methicillin-resistant Staphylococcus aureus. And it's a strain of common skin bacteria that are deadly, right? Because this particular strain has become resistant to antibiotics, and hence the name MRSA, right? Basically describing that it's resistant. And it's believed that, antibiotic resistance is sort of fueled by the overuse of antibiotics. And so it says here that resistance to methicillin occurred in just 1 year, right? So it can happen pretty quickly, okay? And again, a problem if these bacteria that normally wouldn't be deadly can become deadly because they are so resistant to antibiotics. Okay, so it says understanding evolutionary biology is helping fight the superbug. So again, understand the sort of relationship between something very sort of relatable to the medical field, like antibiotic resistance, and how that relates to evolution, right? And again, it's evolution on a smaller scale, but it really is evolution. It really is natural selection. Okay, so like I said, the chapter focuses on evolution on a smaller scale within a population. Okay, so first, I guess you have to make sure you define a population, a group of organisms of a single species that occupy a particular area at the same time. Okay, and obviously there's diversity that exists among members of a population. Otherwise, it's not going to be a very successful population. So there is a specific study or specific field of study called population genetics, right? So basically, geneticists will study this population or populations, right, in terms of the diversity that exists there, right? So they'll look at allele differences, right? How many different types of alleles exist in that population? What's the percentage of those alleles? And again, they can evaluate the diversity of that population and maybe look at it at time point A versus time point B, right, later on, to see how genotype and phenotype frequencies are changing over time. And that helps them assess how much evolution is occurring and how much that population is changing, okay? And so when we're talking about this population genetics and what these geneticists do, think about evolution as a change in the allele frequencies in a population over time. So remember, we talked about alleles before, whether you have that dominant allele versus a recessive allele. This is, again, particularly important if you think about genetic disease, right? If a geneticist is studying some particular human population and they see that the percentage of that disease allele is increasing, there may be a reason why, right? And it would be important to know what that reason is. Our example that we're going to give right now, when we're sort of talking about this population genetics and how they sort of study these changes in the allele frequencies, is not going to be a disease example, but think about that to kind of keep things in perspective. Okay, so again, I mentioned this, right? This field of biology that basically studies these diversity of populations. Now, I also said this in the beginning too, that this is really focused on population on a smaller scale. Okay, so hence the name here, microevolution, right? So this is evolutionary change within a population, right? It's not this long-term evolutionary change like we were talking about in chapter 15 when we're talking about Darwin's theories, right? When we're talking about over basically, the origin of the earth until now in terms of how all these species arise. Now we're talking about one specific population and looking at the gene pool, right? The alleles that make up that population. Okay, so you call that gene pool. And so you can describe that gene pool, kind of like I said on that last slide, in terms of genotype frequencies. Okay, so meaning are you heterozygous, right? Heterozygous, are you homozygous dominant or homozygous recessive? Or you can just look at those single alleles, right? How many dominant alleles, how many recessive alleles are there in a given population, okay? All right, so again, I told you our example is not going to be a disease example, but it is this peppered moth. And again, we mentioned this peppered moth in our chapter 15 lecture as well. So here you got your phenotypes, right? You can see that these guys are dark gray, right? Both the homozygous dominant and the heterozygous. And then if you look at the homozygous recessive, these guys are peppered, okay? Again, this is just sort of review of our genetics chapter, okay? So we should know all these terms, we should remember all these terms. And if we talk about the alleles, remember the alleles are these, right? And what do they stand for? Basically the version of the gene on the paternal chromosome or the version of the gene on the maternal chromosome, right? And so if you're homozygous dominant, you have a big D from your father and a big D from your mother, again, at that particular gene locus. And so what we're looking at is the gene that's going to essentially code for the color of the moth, okay? And so again, you can see heterozygous, right? One big D and one little d. So just remember that when we're talking about alleles, it's just essentially what you got from either your mother or your father. And always you have two different alleles, right, for each gene. Unless we're talking about, remember those the X and the Y chromosome, right? There's some instances where you only have the presence of one allele because some genes are not present on the Y chromosome, but we're not talking about that. So we are always going to remember that we have two alleles. Okay, now we talk about these allele frequencies. Again, remember, this is just a way that geneticists study populations and whether these populations are changing over time, right? Or you can say if they're changing over time, then that population is evolving or evolution has occurred. Okay, so allele frequencies are the proportion of each allele within that population's gene pool, right? So all the alleles that are there, what proportion of them are dominant and what proportion of them are recessive, okay? And so if you talk about the frequencies of the dominant and the recessive allele, they have to add up to 1, obviously, right? You can only have the dominant allele and the recessive allele. There's only 2, right? So this relationship is described as P plus Q equals 1. Okay, I know, oh, sorry, I didn't mean to do that. I know it's math, but it's pretty simple, okay? So again, P is the frequency of one allele, and Q is the frequency of the other. Again, there's not 3 alleles. Remember, there's only 2 alleles, okay? One from your mother and one from your father. All right, so if microevolution occurs, or if you have microevolution, it involves a change in these allele frequencies within the populations, okay? So if those frequencies change, then that means evolution has occurred. And for the most part, there's usually evolution occurring within a population to some degree, okay? So, all right, so it says if the gene frequencies do not change over time, then microevolution has not occurred. Again, for the most part, you do see these frequencies changing over time. And if a geneticist is studying the population, they're really trying to find out in terms of kind of to what degree, right? And then understand what's happening. Okay. All right. So if we look here, this is called, and I'll mention it, I think, on the next slide, but this is called Hardy-Weinberg equilibrium. Okay. So I'll come back to that, but just look at this. So this is essentially what we're kind of talking about in terms of, again, this is a way that these geneticists study this population, okay, to determine if evolution is occurring and to what degree, right? All right. So you can see here, here's your, your population of moths, right? There's 25 moths, 50 alleles. And so you can count them up, okay? So again, you're just counting up the number of alleles, okay, within this. Okay, so you can count up these number of alleles, and you can see here that, again, P is going to equal the frequency of D, right? How many big Ds you have, right, in your moths. You can count them up. Q is the frequency of little Ds, how many recessive alleles you have, okay? And so here you have, for P, 10 out of the 50 alleles are 0.2, right, 20%. And for the recessive allele, you have 40 out of the 50 alleles, okay, or 0.8 or 80%. And obviously, it always has to add up to 1 because, again, you only have these 2 different alleles, okay? See how this works? So, again, you have here the frequency of the recessive allele is 0.8. The frequency of the dominant allele is 0.2, okay? Now, I expect that you guys can understand this, okay? Here now, if you're looking at genotype frequencies, this equation is a little different, right? But again, it still makes sense, but I'm more concerned that you understand this one, the P plus Q equals 1, okay? Here, again, it's not just looking at just the individual alleles, it's actually looking at the genotypes, right? So little p here is the frequency of D, we already said that, right? Q is the frequency of the recessive allele, little d, and you can basically figure this out here. Again, p squared is going to be the frequency of being homozygous dominant, right? The frequency of being heterozygous is this. So as long as you know how many or what percentage of alleles you have, you can figure this out as well, okay? I don't expect you to memorize any of these equations or anything like that. I just want you to understand this, okay? So again, this is allele frequencies, and this is genotype frequencies, okay? So you can basically determine that from that information. Okay, so what is Hardy-Weinberg equilibrium? Again, something you should just be familiar with. I'm not going to ask you, like, give you some, numbers of a population and ask you to, do all these calculations or anything like that. So Hardy-Weinberg equilibrium basically states that allele frequencies in a population will remain constant, okay? And again, this is not really the case, right? So if a population is in Hardy-Weinberg equilibrium, it means that there is no evolution occurring, okay? And again, I told you for the most part, evolution is occurring, okay? So all of the allele frequencies in a population will remain the same, okay? Assuming that there are no mutations in that population, no gene flow, there is only random mating, there's no genetic drift, and there's no selection. Again, in reality, these things are going to be happening, therefore allele frequencies are going to change. Again, remember that this is just a tool that geneticists use to study a population in order to assess how much change is going on, okay? Or what's changing, and then what that might mean, okay? All right, so just to kind of go on with this a little bit more, this basically corresponds to those equations that I just showed you before, right? So if you look at this, and you go back and look at this, right? Same thing there, okay? So you can add that up. So again, you're just multiplying them here, okay? So don't worry about this. I'm not asking you to do Punnett squares along with this or anything like that, okay? Again, it's just understanding that P plus Q equals 1 and just really understanding the concept of Hardy-Weinberg equilibrium and what it's used for, okay? All right, so again, it says these required conditions that I just listed to you, on that other slide, they are rarely, if ever, met, okay? There is always, mutation. There's always changes that are going on in a population, and there is always evolution occurring, really, to some degree, okay? So again, it's just a tool for them to analyze how much, okay? So analysis of the allele changes in populations over time determines the extent to which evolution has occurred. All right, so here's a little bit more here, just kind of showing you this microevolution, and again, showing you kind of what we just talked about here. Here's our, our original population that we saw before, right? Our allele frequencies and our genotype frequencies. And so it says here, if random mating, no selection, no migration, no mutation, then basically, look, it stays actually exactly the same, okay? Again, does this happen in reality? No, not really, okay? If, say, there was just some non-random mating, which means there's particular genotypes that will mate together with each other for various reasons, maybe just because of appearance or what have you, right? So maybe the charcoal-colored moths only mate with each other, and they don't mate with the peppered moths, right? Then you can see that there ends up being a change in genotype frequencies here, but there is no change in the allele frequencies, okay? If something like natural selection occurs, then basically you see, so if selection occurs, you'll see that these allele frequencies will change, okay? This is a typo, okay? Oh, no, it's not. It flip-flopped. Sorry. I was going to say it's supposed to change. If you look here, right, these allele frequencies have changed, and so have the genotype frequencies have changed, okay? So again, take-home message here. If the allele frequencies change, right, so from like time point A, right, the F1 generation to the F2 generation, or time point A to time point B, you see that the allele frequencies have changed, that means that evolution has occurred, okay? If just the genotype frequencies change, then technically evolution hasn't occurred. So in order for evolution to occur, again, for these population geneticists to basically sort of calculate and observe that evolution has occurred, you must see a change in allele frequencies, okay? And again, then they will then further assess that information to see what is happening and how much it's changing, okay? So again, this just basically sums up what we talked about a little bit more. This is showing you the Hardy-Weinberg conditions, and you should know these, right? And then showing you if there's a deviation from that condition. So here again, if it's completely random mating, that's the Hardy-Weinberg condition, and if there's a deviation from that condition, there's non-random mating, then you'll get a change in genotype frequencies. That doesn't really lead to a change in evolution, okay? But however, if you have any of the deviations from any of these other ones, right, that you have selection occurring, there's mutations in the population, there's immigration or emigration, you know, in this case, moths moving in and out of that population, or you also have some genetic drift, and we're going to talk about that in the next couple slides, then there's a change in allele frequencies and evolution has occurred. Okay, again, take home message. If allele frequencies change in a given population, over time, it indicates that evolution is occurring. Okay, Hardy-Weinberg equilibrium and these Hardy-Weinberg conditions and those equations that I showed you are a method that population geneticists use to study populations and to determine if evolution is occurring and how much evolution is occurring, how much that population is changing, and then they may, like I said, then analyze that further from there. Okay, so if we talk about causes of microevolution, again, these are things that we talked about already. So genetic mutations, right? You have to have mutations or variation in the population in order for natural selection to occur, okay? So that's what this is saying here. Again, some mutations might be more adaptive than others. Some are good, some are bad. So it says here, genetic mutations affecting pigment color in those peppered moths have provided the variation needed for natural selection to occur, okay? So again, if that genetic mutation that never caused that you know, change in color never happened, you never would have had natural selection selecting for those different colors, okay? The genetic mutation causes the variation. All right, so again, gene flow, gene migration, this was mentioned before too. This is just movement of alleles between populations, right? So if you talk about gametes or seeds in plants are carried, you know, from one place to another by an animal or what have you, that's gene flow, okay? Breeding individuals will migrate in and out of the population, gene flow, okay? So it says gene flow reduces genetic divergence between populations, okay? So again, the point is that gene flow helps increase variation within the population. It's a good thing, right? Okay, so if migration between populations doesn't occur, the gene pools will become more different over time, right? Meaning that these two separate populations, if they're not interbreeding or, again, if you think about, you know, plants, you can think about it that way too. If there isn't this sort of crossbreeding or between the populations, they will start to become more different, okay? And they could then eventually become reproductively isolated and then thus incapable even of interbreeding, and they could then potentially end up being two separate species over time, okay? So that's sort of the beginnings of these two separate populations becoming two separate species. And we see this happening a lot. So this is showing you an example in plants, right? So here you got a little stream that's separating these two populations, right? And basically you have gene flow across because these seeds get carried by the wind or what have you, and you have this constant flow. And so if you have this flow, these guys are going to remain sort of the same species. If this gene flow didn't occur and you had this geographical separation, over time, these plants would start to become more and more different and would potentially become different species. Okay. I told you we're going to talk a little bit about types of genetic drift, right? Because we mentioned that on the other slide. So what's genetic drift? So changes in the allele frequencies of a population due to a chance event, right? So basically, genetic drift is just something that will remove individuals from a population just randomly, okay? It doesn't necessarily lead to adaptation to the environment. It could basically, and it just, like I said, just sort of occurs randomly. So it says it can cause gene pools of two isolated populations to become really dissimilar because depending on what members of the population die or are removed, it could totally change that allele makeup or even get rid of some alleles altogether, okay? So there's kind of different types here. Bottleneck, if severe interbreeding occurs, when founders start a new population is another areas. And this has a more stronger, or another example, stronger effect in smaller populations. So let's talk about some of these different types that were mentioned there. So
here's what I found.
Sorry, I hit my watch. So here, right, this is showing you a really small population of frogs here, okay? And you got 1, 2, 3, 4, 5, 6, 7, 8, 9. You got, right, 9 green frogs and 1 brown frog there, okay? So there's some natural disaster that occurs, and it kills the frogs, right? However, it only just kills off 5 green frogs, and that 1 brown frog remains intact. Now you can see that he was 10% of the population before, now he's actually 20%, right? So it changes that allele or allele frequencies essentially, okay? So again, this is just like some natural disaster, some event that caused these frogs to die, or a certain percentage of the population to die. If you talk specifically about bottleneck effect, okay, it's a random event that's going to prevent a majority of individuals from entering that next generation. That next generation will be composed of alleles that just happen to make it, right? This is kind of what we're showing here. And again, it's mostly due to natural disasters or habitat loss, right? So again, genetic drift is the more general term. Bottleneck is a specific type, okay? Well, that's an example of bottleneck, really, right there, okay? Founder effect is, again, a type of genetic drift where basically a new population is just started from a few individuals. So it's not a natural disaster that's going to wipe anyone out. It's a certain few new individuals that go, move, and start a new population. And so the only alleles that are going to start that new population are those ones that were carried by those few individuals, those founders, okay? And so that gene pool, that small population, is going to be much different than the gene pool from that larger original population, okay? Again, if you talk about, the reduction in population size, right? So if that population size gets really small, that new population, then there's going to have way more greater effects on allele frequencies, okay? So it says here, formerly rare alleles will either occur at a higher frequency in the new population because those rare alleles are the ones that went to start the new population, or they may be absent, okay? It says here, higher than normal occurrence of inbreeding, right, will occur. Obviously, because if you just had these few people that went to start that new population, you're going to have a lot of inbreeding that's going to occur early on. And a lot of times you'll see more of these rare recessive disorders emerge, okay, in these inbred populations. Okay, so it's showing you here consequences of bottleneck and founder effects with marbles here, right? So here's your original population, your original gene pool. And 1 marble basically is going to equal 10 alleles, and the different colors represent the different alleles, right? And so you can see here, right? So here's your gene pool here, 3,800 alleles, and now something happens. You only have a few members that survive, or a few members go and start a population, and you can see here you don't even have any of those. You have a lot fewer here, right? So only 90 alleles, and then they'll start to breed, and here's kind of what you're left with here. And so if you look at the changes in the percentages here, you can see how this has changed a lot, right? You don't even see the yellow anymore, okay? And so these must be, they're not just looking at one, obviously we're not just looking at 2 specific alleles here, right? So, I forget. I guess I gotta look that up. Anyway, don't worry about it. It doesn't matter. So even if you think about this as different phenotypes or even different genotypes in that population, you can see that one of them is being completely lost in that new population. I'll get back to you guys when we review on this in terms of exactly what those colors are representing. So anyway, like I said, you can see this new population is much different than the old, okay? So again, it's not just one trait and two different alleles that we're looking at here. There's 4 representative colors. Okay, so I'll clarify that. Okay, so non-random mating. Again, we're all talking about these Hardy-Weinberg, criteria, right? And we're just clarifying each one of them. Okay, so individuals don't choose mates randomly. Okay, I kind of told you that in our peppered moth, maybe it's just those gray moths mate with the gray moths and they don't mate with the peppered moths. Okay, and it could be because of various different characteristics. It could be, visual characteristics or physical, but it doesn't only have to be that. Okay, and so it will increase the frequency of homozygotes for a certain loci, right? If you have this sort of non-random mating, and it can play a role in evolution. So here, in this specific island here, non-random mating result in an increased frequency of color blindness. Okay, and you can again think of that in terms of a more serious type of genetic disorder where you can see an increased frequency because of this non-random mating. Okay, all right. So again, we clarified all those different Hardy-Weinberg. Let me just go back here just to remind you, right here, these Hardy-Weinberg conditions that we talked about, right? We talked about Hardy-Weinberg and how it's used, and then we talked about each one of those conditions in a little more detail, okay? And I'll get back to you again on the marbles here in terms of what each color actually represents there. Okay, so this next part talks about different types of natural selection, right? So we talked about the idea of natural selection. I'm hoping we all understand what that is. And these are some things that natural selection requires. And again, we've talked about this before, all of this, okay? So it says natural selection favors the variant that is most adaptive to the present environmental conditions. Okay, we all should agree that that's true. All right. So natural selection will result in a change in allele frequencies of that gene pool, right? Improve fitness of a population, and is the major cause of microevolution. And that's what we're talking about in this chapter. Microevolution, evolution on a smaller scale, evolution related to a particular population. Okay, so again, now we're talking about specific types of natural selection. Okay, so it says most traits are polygenic. Variations in that trait result in a bell-shaped curve. Okay, so we're going to talk about that right now. So 3 types of selection that will occur. Directional selection. Okay, so here in directional selection, the extreme phenotype is favored. Okay, and again, this is due to natural selection, whatever those given environmental conditions are, it's favoring that extreme phenotype. That extreme phenotype is more or better adapted for that environment. Okay, so that curve shifts in one direction. So some examples of this: bacteria becoming resistant to antibiotics, right? It's favoring just those that are resistant. Body size increase in horse evolution is another example of this. Okay, and I will show you pictures of all of these. Next one is stabilizing selection. An intermediate phenotype is the most adaptive for that given environment, right? And so the peak of the curve will increase, but then the tails will decrease, okay? So again, you'll see that all of these curves are these bell-shaped curves like we mentioned in the beginning, but they're going to have various sort of different features, okay? So for example of stabilizing selection is human babies. Okay, so low or high birth weights are less likely to survive. The intermediate birth weight is what is favored, okay? What is most adaptive, right? Like I said, I'm going to show you pictures of all these. Next one is disruptive selection. So here you have 2 or more extreme phenotypes that are favored, not just one, and they're favored over that intermediate phenotype. So the curve essentially will have 2 peaks. So there's an example of these land snails in Britain that vary because of this difference in geographic range. So even though they're the same population, there's still a geographic sort of separation or location kind of influence. And so those that are a certain color will survive better in this one environment, and those that are another color will survive better kind of in this other area. And so you see these 2 sort of extreme phenotypes that are favored. Okay, so again, if you look at these 3 types of natural selection here, right, we can see stabilizing, directional, and disruptive. Okay, so here for stabilizing, the peak narrows. You get basically, and again, this is this bell-shaped curve, the peak narrows because the intermediate phenotype is favored, right? Here, directional selection, the peak shifts, okay? And here you basically end up with two peaks because two of these phenotypes essentially are beneficial. So here's our example of stabilizing selection for human birth weight. Okay, so here it's showing you in blue percent of births in the population, okay, and basically birth weight. So if you look at the blue, this is the percent of births at these birth weights here, right? So here's your intermediate weights, and here are your more extreme, higher and lower weights, okay? So much higher frequency of the intermediate, and then again, lower on each end. If you also look at in red, it's showing you percent of infant mortality. And so if you notice, mortality rates are much higher at the low end. Obviously, if you're talking about really extreme premature, babies that are super low in weight, they're going to have a much lower survival rate or a higher mortality rate, right? That's what it's showing you here. So high mortality rate, that mortality rate really lowers here at the intermediate sort of healthy weight range. And then you also see an increase in mortality weight at that higher end. And again, this is due to, other types of genetic abnormalities or developmental abnormalities that lead to this increase in birth weight. And then thus an increase in mortality. Okay, so blue is the percent of births in the population. You can see much more in the intermediate range. And then red is the mortality rate, higher at the low end. And then, and then dips down here and then higher at the high end. Here is showing you directional selection, right? Again, this is in the sort of the evolution of the horse, right? That you basically, so kind of initially, right, this is sort of the body size that's favored. But as over time, basically a larger body size is more favorable, right? And you can kind of see here, you have many more individuals with this larger. So that curve is shifting. That's why it's called directional selection. Here is our example of the disruptive selection, or these land snails, okay? Again, it just happens to be that there are sort of two geographical locations within this population. And so if this guy, the guy that lives in this area, it's this color or pattern on the shell is more favorable, obviously due to probably, prey selecting for them, and also the ability to properly blend in with the environment. Here, if you live in the area that this guy looks like, you're going to be more fit to survive if you have this pattern and this coloring on your shell. Okay, and so you can see again, it kind of started off as sort of this initial distribution like this, but over time you see these two peaks that form. Okay, all right. Another form of, or kind of something that has to do with natural selection, is something called sexual selection. So there are adaptive changes in males and females that will lead to an increased ability to secure a mate, right? Obviously, this is going to affect your sort of fitness within that population, right? So there ends up being, right, males an increased ability to compete with other males for a mate, right? So if they're bigger and stronger, they can fight other males off and basically be the ones that will mate with those females. For females, they choose to select a male with that best fitness, right? The ability to produce surviving offspring. And so that's going to obviously affect that population, okay? So just to kind of go a little more with that, something called female choice, right? So the choice of a mate obviously is pretty important, and especially in certain species too, because and more important to the female because they produce fewer eggs. For the male, they're producing lots and lots of sperm, but for the female, there's a limit, right? And so the idea is that females are really the ones that are sort of directing this and choosing the mate, okay? So it says here, for this good genes hypothesis, females choose mates on the basis of traits that improve the chance of survival. This runaway hypothesis, females choose mates on the basis of traits that improve male appearance. There's something called sexual dimorphism, right, where you see males and females will differ in size and in other traits. You see this all the time in different species of animals, right? So if you talk about birds, right, so female choice explains why male birds are more ornate than females, right? And so and this is kind of a bad example, I probably should have a real picture of real birds, but you can pretty much see, here's your female, kind of boring, right? And here's your male with these very colorful and sort of ornate feathers, right? And that's because that's going to attract the female, Okay, and so again, this is the sexual dimorphism, meaning, difference between the male, the female, and the male, and in terms of attracting mates and what they look like. Okay, again, this is going a little keep a little further with this, and I kind of mentioned this a couple slides ago, but male competition, right? And so they will basically, compete with other males to basically inseminate as many females as they can. Okay, and so it's kind of a cost benefit analysis here, right? It says, is access to mating worth the competition, right? Like, is it really important to that male to engage in some sort of fight to secure this mate when potentially they could get hurt doing it, right? And so that's what this is saying here. And larger males will need more food; they're more likely to get hurt, right? So is that really worth the sort of increase in mates that they will obtain? Okay. And obviously you've heard about this before, that, these males can become very territorial, right? And basically defend that territory against predators, but also maybe against other males that are trying to mate with their females, right? And so this is kind of showing you an example of the behavior, to basically scare away that sort of threatening other male that's coming along. Okay, and you can see this here too. Again, this kind of warning call, and then these guys engaging in a fight for whatever reason to sort of defend their women, right, or their females, or their territory. Okay. Again, I think we talked about this a lot too. This is the last part of this lecture. So maintenance of diversity. So it says populations with limited variation may not be able to adapt to new conditions, right? Again, you need to make sure you have a certain amount of variability within that population that is going to be advantageous to that population. Okay. All right. So it says natural selection causes imperfect adaptations, right? Again, I told you this, right? There's mutations, there's genetic changes that exist, within that gene pool of a population, and for whatever is more suited to that environment is what's going to be selected for over time. Okay, it's not like it has a sort of perfect sort of end goal for a certain species, and it's always selecting for the most perfect individuals within that species, right? There's times when a certain environment selects for certain individuals, and then that will change, and maybe those individuals that were selected for at that certain time point actually then now again are not suited to that environment, and then they may die off, right? So if you look at evolution of a certain species, you don't just see this perfect kind of newer version, every so often that forms. You see different species that form and then die off, right? Lots of species become extinct, and again, because it's not something that's planned, it's just that environment selecting for whatever individuals are most suited for that environment at that given time, okay? And so this is all what this is kind of saying here. It says the benefit of freeing our hands outweighed the cost of spinal injuries from assuming an erect posture, right? So we walk upright, our spines really still haven't evolved perfectly to being able to do that, so we have issues with the spine and back issues, right? But it freed up our hands and allowed us to be able to use our hands. And so if that didn't happen, we would be at a lot different place than we are today. Okay. So that's just an example there. Okay. I think we talked about all of this. Oh, the one thing is that, yeah, so it says here, so this is kind of talking about the existence of subpopulations. And again, you can kind of think of the existence of subpopulations as, as being another illustration of diversity, right? Or variation within a population, okay? So you basically have this population, but again, there's these sort of subspecies that exist within, and that's basically displaying that variation. And then over time, those subspecies may actually become a separate species, right? So we gave an example of beak size, right? And those Galapagos finches, dependent on the availability of food supply. Again, it could be not just food supply, other types of variations could be due to the environment, right? And the existence of different colors, right? In terms of blending in the environment, we talked about things like that before. So here's an example of that. Right, and so again, it says subspecies help maintain diversity. So you can see these are all subspecies of this particular species of snake. They are not different species themselves, and the color coded here corresponds to the geographical region that they live in. Okay, and so you could see that they're all separated geographically, okay? Now, they're subspecies, they still have the ability to interbreed, right? So they haven't become separate species. However, over time they may, right? And so, again, it's just their changes in appearance that we're showing here is based on where they live, okay? And so again, that's a really diverse population, okay? Or, you know, and it's even, and so I guess you have to have these sort of localized variations, I guess, to basically consider these subspecies, right? But it's a large amount of variation within that population. Okay. Here's another thing, or another reason why diversity is important, right? Or variation within a population is important. Okay. So it says up there recessive alleles. We know what recessive alleles are. It says heterozygotes will shelter recessive alleles from selection, meaning the heterozygotes that have these recessive alleles, and if you think about it as being like a recessive, disease or something like that, they don't have the disease, but they still carry these alleles around, right? So that's what this is saying. Heterozygotes will allow even lethal alleles to remain in the population at low frequencies forever, right? Because it's not really affecting them. It's not making them less fit. So again, it assists in the maintenance of genetic and therefore phenotypic variations in future generations, because again, something may change that then does make it beneficial to have that recessive allele. And so if that recessive allele went away, right, and we had that lack of variation, that could be a potential problem. Okay, so there's something called heterozygote advantage, right? So the heterozygote actually can become favored over the two homozygotes, again, depending on what environment you're in at that given time. And so here are two examples, right? So cystic fibrosis. So carriers of cystic fibrosis are actually protected against typhoid fever. Now, if you're in an area where you're never exposed to typhoid fever, it doesn't really matter, right? And so you may see this sort of, then there really isn't a heterozygous advantage, right? But in certain areas where this is helpful, it is super important, right? Sickle cell disease is another one, okay? Heterozygous individuals for sickle cell disease don't die from sickle cell disease, right? They don't die from sickle cell, but they have symptoms, and they don't even have symptoms, right? They're heterozygotes for sickle cell. So they don't die from sickle cell disease, but what also happens is that they don't die from malaria. So it protects them against malaria. And again, in areas where malaria is prevalent, this is an advantage, right? And so this is why why it's important that this recessive allele has been maintained, right? So it says the sickle cell anemia allele is detrimental in the homozygote, right? If you're homozygous recessive, you have sickle cell disease. However, if you're a heterozygote, you're okay. You don't have sickle cell disease, right? And you're actually more likely to survive malaria, okay? And so you actually see the sickle cell allele, that recessive allele, at a much higher frequency, like I said, in malaria-prone areas because of this protection that it serves, okay? So it says this is a form of stabilizing selection, right? Again, it's that intermediate phenotype, that heterozygote, that is favored, right? And again, you're only going to see this higher frequency or that favoring in areas where malaria is prevalent. Okay, so this is kind of just showing you, you look here, right? Areas where malaria is prevalent in yellow here, where you see sickle cell, and then where you have blue, overlap of both. Okay. And again, it's the overlap is sort of indicating that you have you know, particularly these you know, more of these individuals with heterozygous genotypes, because that serves as a protective agent. Okay. Oh, and that concludes the presentation. So like I said, we'll review this next time in class.